Hi everyone, my name is Claire Rogers, and today I'll be explaining my poster on designing the L452R variant of SARS-CoV-2 mRNA vaccine. The SARS-CoV-2 virus has mutated several times, which has created many different variants throughout not only the US, but worldwide. I designed the vaccine for the L452R mutation in the novel coronavirus based on the vaccines already available, like Pfizer and Moderna. This is significant because the specific mutation is prevalent in the United States in the California region. Studies have shown that this is potentially very infectious and has a high infection rate, which means that it could more likely affect those in the United States and lead to serious and long-term effects. So if we look over here in the background, we can see the whole SARS-CoV-2 virus. You can see the viral RNA is here in the middle, enveloped by a membrane and surrounded by these spike proteins. So the spike protein can take on many conformations, one being the pre-fusion conformation here in purple and the post-fusion conformation here in pink. So these different conformations are important because it affects how likely the virus is to infect a host cell. So the pre-fusion state exposes the RBD, or receptor binding domain. This is where the virus will attach to its host cell and switch to the fusion state. The fusion state then injects all of its viral genome into its host cell and then quickly changes into the post-fusion conformation. This is why we want to find a vaccine that attacks the pre-fusion conformation, because then it stops everything from being infected and stops the viral genome from replicating. And you can see the pre-fusion conformation here at the bottom. And in order to keep it in the pre-fusion conformation, we need to have the two proline mutations in 986 and 987. So the reason that proline keeps it in this position is because it is cyclic and helps it from interacting with other parts of the spike protein. So moving on to my results, we have the plasmid map. For the plasmid map, I chose to use ECO-RI because it's in the multiple cloning region. This helps linearize the spike protein in order to go through 2 rounds of mutagenesis. So the spike protein map looks something like this. You can see we have our N-terminus domain, our receptor binding domain, spike protein, and our mutations. My specific mutation, the L452R mutation, is present in the RBD, and the 2 proline mutations are shown here in darker red. And our mRNA map is shown here. We have a T7 promoter, a 5' UTR, which will insert in C3, our spike protein, a 3' UTR, which will insert at ASL, and our poly-A tail. Moving over to C, we have our primers. I showed the forward and reverse primers that will be used for each mutation. We have the lysine to proline and valine to proline, and then leucine to arginine, all shown there. Then we finally have our pimal structures. I labeled the 2 proline mutations in red, you can see here in the middle, and the L452R mutation is shown here in blue. The approach to our experiment is to insert the L452 mutation into an mRNA vector using plasmid DNA to replicate. And the aim of the study is to design an effective vaccine for the SARS-CoV-2 with the L452 mutation. This is important because if the L452 mutation decides to emerge and be more potent, then you have a design for the vaccine. And then future direction, after the template is created, you will use in vitro transcription and HPLC purification to ensure that the vaccine is correctly made and good to go. My acknowledgements go to Gabrielle Perkins and Dr. Roberto de Guzman.